Wielding the power of DNA will result in opportunities worth trillions of dollars if you know where to look. Welcome to today's webinar, The Awesome Power of DNA. Pink pineapples, personalization everywhere, and sensing all the things. My name is Rachel Darasek. I'm the Vice President of Marketing and Product Strategy here at Lux, and I'll be your host for today. Our presenter is Sarah Olson, Director of Research, who leads Lux's Food, Health, and Wellness area of expertise. Sarah, tell us a little bit about it.
Sure, happy to. I come from an agricultural background. My PhD is in biochemistry with a focus on crop genomics. So the topic of power that DNA has is very near and dear to my heart. Over my career here at Lux, I've had the chance to work closely across our agricultural expertise as well as food and health and consumer aspects of technology. So this topic is certainly one that touches all of those topics as well.
Thank you, Sarah. I'm quite sure everyone listening right now is eagerly awaiting your insight based on that background. And having participated in your rehearsals, I can confidently endorse that anticipation. But before we get there, I wanted to provide a little bit of context about why Lux is here today talking about this topic. So let's start with our members. You work with them every day, Sarah.
Sure.
They have a singular focus, driving growth through tech innovation for their organizations. Everyone who's listening right now knows that tech-driven disruption is nothing new. Horses gave way to cars, telegraphs to telephones to smartphones. Insert your favorite technology here.
Plows into tractors.
Plows into tractors. What is new, however, is the accelerating rate of innovation that we are experiencing right now. The average lifespan of an S&P 500 company today is less than 20 years. You compare that to the 1950s, the average lifespan of an S&P 500 company was 60 years. It's not that long ago.
Mm-hmm.
The accelerating rate of innovation translates to a more individual level as well when we think about how it affects us on a day-to-day basis. As an example, one client recently shared that they're expected to advance 2 ideas per year to a certain late stage in their funnel. That's a huge shift from what they had been expected to do in the past. Another client told us that what would take them a year to investigate now have to do in 3 months.
Wow. Not a lot of room for error.
No. And it isn't as if more time or more resources magically appear to help our clients meet these expectations. In fact, many times the opposite is true, and especially in the world of CPG. Operational considerations, cost, channel growth, regulatory pressures, and supply chain complexities are all creating really significant roadblocks. And adding insult to injury, yes, there's more. The only thing accelerating more quickly than internal expectations are the external factors. We think about consumer preferences, organic, local, non-GMO, food for health. These preferences seem to change overnight. I know mine do as a consumer. Then you start thinking about the landscape, new startups, new products, new markets. They seem to be emerging almost daily. Add in the tsunami of information available out there, concept of digital transformation, which is not just a concept, but an actual reality. Just talking about all these things, I'm feeling a little bit overwhelmed. I can only imagine how you and the folks you talk with every day are feeling.
It's a stressful situation.
Since failure is not an option for any of us, the question is, what do you do to succeed in this environment? How do you decide what technologies are right for your organization to prioritize, who to work with, when to act, and how do you do it quickly and reliably? That's where Lux comes in. We partner with innovation executives at global Fortune 500 companies in physical industries like materials, energy, food, health, and wellness to help them make the right decisions faster. We use advanced analytics techniques such as data science and machine learning to track all the information. We curate the data based on our domain knowledge, and then we pair it with expert insight to help you make sense of it all. So with that, let's turn our attention over to Sarah, who's going to give us her Lux take on some obvious and not so obvious opportunities in wielding the power of DNA. At the end of the webinar, we'll take a few questions. If you'd like to submit a question, please use the Q&A box on your screen, and you can enter your question at any time. No need to wait until the end. Sarah, over to you.
That is the perfect segue. All righty. So, as I alluded to a little bit previously, the awesome power of DNA really touches so many different technology areas, so many industries. It is a mouthful to talk about pink pineapples, personalization everywhere, and sensing all the things, but it really is indicative of just how broad this power is, and it's part of the reason I enjoy talking about this so very much. It's also a topic that seems to be very accessible to a lot of folks in pop culture. Um, Messing around in DNA and genetics is a favorite of Hollywood directors everywhere. Um, and I'd like to talk a little bit about how the movies get it wrong, because I think that contributes to some of the challenges that we face in this space. So whether it's Jurassic Park and their obvious misinterpretation of molecular biology, to even the way Bond villains have tried to and failed to scientifically make use of the power of DNA, there's a lot of imaginings of how this might work, and they're just not correct. Um, there is one case I'd like to talk about that gets closer to scientific accuracy, even though the scenario is a little bit far-fetched. We've called Gattaca is based on a premise of a future society driven by DNA sequencing to identify people and the ability to tailor the genome of a developing baby in utero to avoid various diseases and so on. So it's a fascinating film. I really enjoy the storyline as well. If you haven't seen it, you really ought to. And one of the reasons it's such a great example for today is that it's not actually scientifically beyond possibility. It's just that there are some significant barriers, both technical and regulatory, that stand in the way today. So I mentioned that the future scenario relies on DNA sequencing to identify people essentially at the drop of a hat. And sequencing is just way too slow to achieve that scenario today. It's on the orders of hours to days to sequence a whole genome, not seconds. Now you cannot legally edit people or embryos for preference today, although as of now, here we are in November of 2018, and we just very recently had news of some gene-edited babies being born in China. It's a situation that is still very actively unfolding, but it certainly was too recent to make the press's release slides. To add some challenge there as well, in most cases, we don't really know which genes lead to which diseases. There are some specific single gene disease cases, but for the most part, it's actually a complex genetic makeup, and it's hard to understand and predict. So Gattaca's vision of the future is scary, but it's actually inaccurate. We'll get to more a little bit later on, as Rachel, as you had mentioned, on some of the more accurate future predictions. Before we get there, I also just want to mention that there are some legitimate ways for using the power of DNA to go wrong, and I don't mean in the Jurassic Park sense. So pictured here is a gentleman named Athanasius Theologists. This is the developer of the Flavor Saver tomato, a tomato that doesn't ripen and then rot in the span of, I don't know, 24, 36 hours sitting on the kitchen counter. It was a really ingenious approach combining DNA from other organisms into the tomato genome. Unfortunately, due to some misunderstandings by consumers and those in the popular press, the product was panned and had to be pulled from the market. We have the cautionary tale from PepsiCo's work with a small company called Genomics, attempting to use human taste receptor cells to develop better and tastier non-calorie sweeteners. And due to a severe misinterpretation of the facts, some folks were led to believe that Diet Pepsi was actually including aborted fetal cells as a sweetener, which is horrific and absurd, and anyone with a technical background knows that to be false. But nevertheless, it did impact PepsiCo significantly, and the partnership hasn't been as productive as it could have been as a result. And the other example that I like to highlight as a cautionary tale is the case of Roche purchasing 454. This was Roche's attempt to really buy into the sequencing space in 2007, at a time before there was really a clear dominator in the space, and they spent over $150 million. They made sort of an audacious bet, And we could have told them at the time it was a bad idea. They didn't ask. And instead, 6 years later, they shut down that side of the business and they had to let hundreds of people go. And it was a real loss for Roche. I don't want to bore you with all the details right now, but it was very easy to see this wasn't going to go well if you had taken the Lux lens to things. Which brings me back down out of these crazy scenarios to the reality that there are actually legitimate opportunities to wield the power of DNA and have some success. We're going to talk first about some pretty straightforward and linear opportunities. These are sort of near-term things that you can start working on today. And then we're also going to put on our Hollywood writer hats and look at the future and think about some things that are a little less linear, but could still be pretty important aspects of the power of DNA going forward. So, starting with some of those linear opportunities, they really align across the food and health value chain that we cover in great depth here at Lux. The team I get to work with on a daily basis is covering these topics all the time. They live and eat and breathe the industries that these linear opportunities come from. And those are really starting from the farm, production agriculture, the ability to make ingredients from crops, the making of food products from various ingredients, and then those impacts on consumers and their health. And if you look at these, agriculture, food, and health are trillion-dollar markets. It's not very often I get things where the number starts with a T. Millions and billions and so on are fine, but these are trillion-dollar markets. And what it means is we have three different trillion-dollar, multi-trillion-dollar markets being impacted by the power of DNA. So if you're not paying attention here, you're already losing money. If not today, you're setting yourself up for even bigger losses and missed opportunities in the future. I just want to highlight a couple of examples. Let me start with one example from the farm. Again, near and dear to my heart and my own personal background. You can use the power of DNA to make the impossible possible, to make a crop or a variety that didn't exist before. You can also use DNA to speed up what are otherwise very slow and sometimes painful breeding processes to get to better traits and crop improvements. One great example of using the power of DNA is the pink pineapple developed by Demonte. This is a pineapple that is relatively normal in all respects, but expresses a gene for lycopene, a nutrient usually found in red fruits like tomatoes and so on. It's an added nutritional benefit for pineapple that wasn't possible in nature otherwise. We also have an interesting story of using CRISPR, a genome editing method, happened earlier here in 2018, to domesticate a wild variety of tomatoes in a single generation using this multiplexed CRISPR approach. And what that actually did was allow the developer to demonstrate a net savings of $100 million in 9 years in product development, doing that single generation instead of the sort of old-fashioned way, if you will. There's a couple of strategic imperatives here if you want to go in this direction, and they're pretty straightforward, but I think they bear discussing. The first being, some of this is unsexy. So do homework. A whole genome sequence that is robust and thorough for the crops that your business relies on, that is your ticket in the door here. You cannot wield the power of DNA in the ag space without that. The other thing is some folks come to this area thinking they're just going to buy a variety that's been developed, and theoretically that's fine, you can do that, but you're going to end up paying market rates for something that's close to, maybe not exactly what you want. You're better off building or co-developing a platform for trait development in the crop or crops that are most relevant to you. If we look a little further down the value chain and look at using the power of DNA to develop new food products, this gives me a chance to talk about another topic that sounds like science fiction, cellular agriculture. So cellular agriculture is a method that goes by a lot of names. You might have heard it called lab-grown meat. Artificial meat is a name that has been thrown around a little. Recently, US developers have gotten together and started using the phrase cell-based meat. I detest that one. I don't think it doesn't stick. But having said that, by any other name, this is the idea of using technology and DNA-enabled approaches to create foods that either are made of animal cells or mimic animal tissues without actually involving the slaughter of whole live animals. We have assessed this area, and we've used a method called the Lux Innovation Grid to represent our research findings. So the Lux Innovation Grid it is a bit of an eye chart here, I understand, but just very, very briefly, this ranks companies in a given technology space on the basis of their technical value on the y-axis and their business execution on the x, giving you a nice understanding of where companies sit in terms of their skill in technology development and how they function as businesses. And what we can see in this particular Lux Innovation Grid is a collection of companies in the top left or high potential quadrant. These companies are promising technologies, but they're not there yet in terms of being legitimate sort of producers on the market or being at scale production. And that really describes this area. There's a lot of promise. There's a lot of technology in development that we think is exciting. Most of these companies, though, are not ready to be your JV partner. They're not ready to supply you with 1,000 tons of product that you're going to turn around and take to market. Our recommendation here is much earlier stage than that. Identify the startups, potentially even from that group listed there, that are ready for a Series A round and make an investment on the basis of either your long-term financial gain or the strategic imperatives that your own organization may have. I think the way Tyson, the meat company in the US, has interfaced with the cellular agriculture space is a great example of how you can get really proactive about being part of the disruption that's coming for your own business. Now, for all the really fascinating and science fiction sounding things you can do with DNA, all of it, every single piece of it, comes back to these same 4 basic skills that you can combine in different ways into incredibly powerful tools. That's the best news, is that it's just 4 basic skills. That's the ability to read or sequence DNA, to analyze DNA sequence data, looking for alignments to existing sequences, looking for markers, and so on, the ability to write or synthesize new pieces of DNA, and the ability to edit existing DNA sequences within cells or organisms. These skills are evergreen, as advanced, as simple to advanced, really, as a tool gets. It's all going to come back to these 4 same skills. Now, we walked through a couple of somewhat straightforward examples, things that if you're already in this space, you're probably already thinking about or at least working on a little. But I also want to think a little further forward and talk about some less obvious future possibilities. These are, nobody has a crystal ball, but if I had my best guess, these are things I think are likely to come from wielding the power of DNA. The first example doesn't have a lot of real market implications today, but I think it's an interesting examination of DNA being used for its other powers, not just to code genes. DNA can be an adhesive. So research from the Weiss Institute at Harvard shows the ability to use complementary sequences of DNA as specific and reversible adhesives. So you can imagine taking pieces of material, coating them in complementary DNA sequences, and then allowing the natural bonding between those DNA base pairs to actually bring those pieces of material together. It's a fascinating potential application. If I were a materials developer, I would be really interested. It's also not something I see disrupting the adhesive space anytime soon, but something to be aware of nonetheless. In another vein, DNA is also a data storage mechanism. So as you think about the future of data storage, you're not going to have a DNA-powered hard drive anytime soon, but DNA is a phenomenally long-lived data storage apparatus that can be used to store any number of pieces of information. The next nonlinear example I want to talk about comes more directly from the food, health, and wellness arena. and that is the growing propensity to want to sense all the things across agriculture, food, and health. To help illustrate this, I'm leaning on a cartoon from Ali Brosh, a cartoonist I think is just fabulous. I love her stuff. The reason this is important to me is that what's coming is ubiquitous, cheap, fast, and accurate point of view sensing. And we're calling this point of view sensing because right now there's not a good name for this in the industry. There's a lot of developers going in this direction, a lot of consumer interest. A number of the CPGs we talk to are working in this direction, but it's a convergence of different areas that's coming together without a unifying name. And point of view feels to us like a very reasonable way to talk about this space. This is being driven by a better understanding of the DNA that is in and on most everything we interact with, and a better ability to measure and read and analyze what DNA is there in any case. There are a few developers that I think are worth highlighting here just to paint a picture of where we're going. So Mammoth Biosciences is using the CRISPR apparatus to develop paper-based near-instant diagnostics for detecting presence-absence of certain DNA sequences. So you can imagine the applications in food safety alone are significant, to say nothing of clinical medical applications as well. Moloko is developing an under 5-minute milk quality assessment. Now that's not based purely on DNA information. There's also aspects of protein content and fat content built in, but the idea of being able to sense at the point of use near instantaneously is becoming more prominent and will continue to become more ubiquitous. You can imagine arriving at a point where, as a consumer, you might sit down to lunch, and before you choose what to order from a menu of personalized nutrition options, nonetheless, that you might make a quick check of the status of your microbiome and figure out, gee, can I have a burger or should I really have a salad, for example. If you're wondering what you should do about it, my best recommendation here is start to think about food safety as a continuous process. Don't think anymore of preparing a batch of product, taking a sample to a lab, waiting hours to days before determining a result. Start to think about how you can incorporate continuous testing and verification within the line of your production. Further, you're going to need to think of DNA as a traceable fingerprint that's in and on everything you touch and everything you produce. It's either going to be an asset or a liability. If you do nothing about it, it's definitely going to be a liability. So with that, I do want to share just a few final thoughts about the Gattaca example that we started with. As I mentioned a little bit earlier, technical barriers are already coming down and regulatory barriers seem poised to follow. So sequencing is getting faster. In, I believe it was September of 2018 here, BGI announced a 3x increase in the speed with which they can sequence human genomes. So that's 3 times faster than the previously accepted rate. It's certainly not fast enough to get us to instantaneous rates, but it demonstrates we haven't hit a threshold that we can't get under yet. In terms of figuring out which genes correlate to which diseases, again, we're not there, but there's a method called polygenic risk scoring that's combining data from millions of markers across a genome to get surprisingly good at predicting various dangerous and even fatal diseases within human genomes. So we are getting closer there. Regulators are also relaxing restrictions on human gene editing. Now, so far that's been in pursuit of clinical trials to treat various major adult diseases, but there's been a very surprising development just this month, November of 2018 here, out of China, where a researcher claimed to have used CRISPR to edit the genomes of 2 human embryos, which were then implanted and led to the live birth of 2 twins whose genomes had been edited. Now, the situation is still unfolding. Obviously, that was done without regulatory approval, and the backlash may sort of ripple for quite some time. But it certainly speaks to the increasing capabilities unfolding here and the tension between technical capability and regulatory barriers. Which brings me to the conclusion, now is the time to act. Do not wait for a nightmare scenario to sort of come to you. Use the unfolding technical advances that you're seeing happen around you to benefit your business today. So I'm not going to leave you with just that. That feels very doom and gloom. Instead, I think it's important to have a bit of a to-do list, something to take this big sort of hairy issue and make it a little bit manageable. We're going to look at things to do now and then things to plan ahead for. First, right now, you need to be thoughtful about every single partnership you could or should be making to facilitate your organization's ability to read, analyze, write, and edit DNA. How are you going to thoughtfully use and incorporate those skills? Also, in every single scenario relating to manufacturing or product development that you are a part of, consider the potential role DNA can play in improving your margin, increasing performance, or, as I mentioned earlier, making the impossible possible. When you think about planning ahead, obviously it's a little bit nebulous. It's a little hard to predict exactly where the future might go, but there are some things that we're pretty confident in here. We're fairly confident DNA will power an ongoing revolution in personalization and point of view sensing across the food and health value chain. Much like the industrial and green revolutions before us and the ongoing digital revolution, the biological revolution is happening now as well. So you need to plan ahead to be ready and to take advantage. And finally, put on, as I mentioned, your Hollywood director hat. Your business is going to be disrupted by the power of DNA, not in the form of engineered dinosaurs or anything like that, but in the form of very real disruptions coming from farther outside your current sphere of influence than you're used to looking. So take a broader and more creative perspective in planning with your organization. As you do strategic planning, it's time to think broader. Otherwise, you will be blindsided. There's no two ways about it. So on that less than cheery, I just want to thank everyone for listening in, and I'm happy to hear the questions our audience has for us.
Thank you, Sarah. These ideas are really outrageous. I do have to say, it seems a bit like science fiction in real life. On the one hand, tons of benefits. On the other hand, lots of scary things to consider and tons of opportunities out there. So I do see some questions coming in. As a reminder for everyone else, please go ahead and type your questions into the Q&A box you see on the screen. So let's get to the first question. So early on in the presentation, you talked about the Roche acquiring 454. The question, and you mentioned that you would have advised Roche to not move forward with that deal. So the question coming in is asking for you to please comment on that a bit further.
Oh, on the why? Sure. That's a very valid question. Simply put, at the time, Roche was looking to buy into a space it didn't really have capabilities in. And the 454 platform was one of a number of available solutions. And it was clear to me and to some others at the time, the 454 platform had some serious shortcomings in terms of both read length and the ability to multiplex, so that essentially the net value of any one sequencing run from a 454 platform was less than that from other competing platforms at the time. Roche sort of seems to have seen it as a way to buy into the sequencing space rather than looking for a technology that might augment existing capabilities they already had in a different way. And I think it's worth pointing out there is an example of someone who did come in and do this well at the time, and that's Illumina. So most folks in the DNA world today are at least familiar with the name Illumina. But in 2007, they were still a very small player. And around the same time Roche bought 454, Illumina made a very savvy acquisition of a company called Celexa, which is where they got the technology for the bridge amplification PCR that forms a key part of its currently market-dominating sequencing platform. So at the time, it was very easy to see how that one technology would fit into what Illumina was already developing, and it was a much smarter choice than what we saw Roche do with 454.
Interesting. So it sounds like maybe Roche made some interesting decisions or didn't quite do their due diligence and ended up having a bit of buyer's remorse at the end.
Yeah, I think buyer's remorse is a good way to put it. I think their failing there was they failed to think big about the role sequencing would play going forward and the importance of having both depth of coverage and read length.
Thank you for that. So that actually relates nicely to the next question that I see here on the list. This name jumped out at me as well, as I've seen it in the news a lot lately. But when you flashed up that Lux Innovation Grid, it included impossible foods.
Oh, yes.
So the question here is asking about how or if impossible foods are using DNA in their products.
They are, and they're doing something sort of fun, and they are making the impossible possible in a way. So for anyone who's less familiar, Impossible Foods is known for making plant-based burgers and meat replacement products that their stated intention is to get as close as possible to mimicking an animal product without using animal products. And the innovation that they have come up with that does use the power of DNA is the way they've achieved the bleeding effect in their burgers. So what they've done is they're using a synthetic biology approach and using microorganisms to produce heme proteins, which are the same proteins that give animal blood the red color, iron-containing proteins. They have that the mouthfeel and taste that you would look for as an indicator of an animal product in a lot of cases. And what's really clever about what Impossible Foods has done is that they didn't go and find a heme gene in the cow genome or the chicken genome or what have you. They actually found the heme gene in a soy genome. And that's the gene that they're using. So they're able to continue to make that plant-based claim and avoid alienating some of their very staunchly anti-animal product consumers while still achieving that animal-like product quality in the end. It's very clever.
So they're avoiding the whole PepsiCo situation that you mentioned.
A little bit, yes. Some of the very diehard vegan consumers take issue with the synbio approach they're using at all, but I think you win some, you lose some at that point.
Absolutely. So let's go to the next question here. You mentioned being thoughtful. That was the language you used about the 4 technical skills that you need in order to wield the power of DNA, as we've been discussing here today. The question here is whether or not the folks on the phone need to have all 4 of those skills in-house in their organizations.
Oh, definitely not. That's a good point to clarify. All 4 skills will be relevant in different proportions for different tools. There will be cases where really all you need to do is to be able to read and analyze sequence data, in the case of a diagnostic tool, for example. So there will be cases where you don't really need all 4 skills, or you need one most of all, but not others. The other thing that's worth mentioning is some of these skills are tougher to build in-house compared to the ease with which you could interact with a service provider or other tech developer that already has that skill. The DNA analysis piece is probably the easiest illustration of that. So to be good at analyzing DNA, analyzing DNA sequence data, I should say, you really need strong computing power and a good ability to build algorithms. Assuming that you already have a solid genome sequence of your organism of interest, like I mentioned early on, was very important to do. Assuming you already have that, it's a little bit reinvention of the wheel to go and build your own computing power and gather your own algorithm development expertise, when instead you might be able to bring your raw sequence data and your genome sequence to a service provider that could handle the algorithm development and the analysis for you, identify markers, do the alignment, whatever it is that you need. So it's worth being thoughtful about whether to do it yourself or partner up, or simply just pay for a service on a case by case.
The classic strategic build-buy situation.
Absolutely.
Thank you for that. So let's get to the last question. I'm getting close on time here. The question here is about sensing.
All the things.
Sensing all the things. And as you know, there was yet another recall of romaine. At least here in the US, this seems to be all too frequent. So that's what I would love to have a little sensor in my romaine lettuce that tells me whether or not I can eat it. But the question here is recommendations of where to get started if someone is interested in pursuing this idea of continuous sensing for food or food products.
Absolutely. Yep. The romaine recall was the scourge of Thanksgiving tables everywhere this Thanksgiving in the US for sure. It's a good question. It's a bit daunting to imagine a complete paradigm shift for an entire industry, and how do you eat an elephant one bite at a time, right? The best place to start, I think, is in looking at ways you can incorporate these sensing technologies to reduce your recall costs. Beginning with inline production. And small sensing approaches that you can incorporate to catch problems earlier on prior to shipping out lots of product is probably the best place. And the reason I think that's the right place to start is because it's the easiest place, or the most straightforward place. I don't know how easy it will be, but it's the most straightforward place to make a case for a legitimate and measurable return on the investment, because you should be able, with some small work, be able to demonstrate a significant reduction in your recall expenditures and your recall associated budget from a relatively minor investment in sensing. So that's really where I would begin as a first step.
Not to mention the bad press.
Oh my gosh, yes. The romaine recall itself, in fact, was significant enough to bring the FDA and the USDA together to agree that going forward, U.S.-produced romaine lettuce will carry a region designation. That is a relatively unprecedented and incredibly rapid step that's going to have some significant impact for romaine producers in the U.S. So some of our ag analysts were just writing about that, in fact, yesterday, because it's very topical and very relevant.
Glad to hear there's some action being taken. So it looks like, unfortunately, we're out of time. I'm sure we could talk much longer about these fascinating topics.
I could.
But Sarah, thank you again for today's presentation, all this insight and recommendations. For those on the phone, we weren't able to get to all of the questions, unfortunately. If you submitted a question that we were not able to answer, you can expect that we will follow up directly. And if you'd still like to submit a question, you can email us directly at questions@luxresearchinc.com. And finally, if you found today's presentation inspiring or interesting, tell your friends, of course. And you can also follow Lux Research on LinkedIn, Twitter, YouTube, or follow our weekly blog for ongoing information and insights across a variety of topics. And of course, you can always contact us to learn more about how we can help you drive growth through tech innovation for your organization. Thank you for joining us.